I think we'll go ahead and get started so we can stay on time this morning. My name is Monica Farley. I'm the president of the SSCI for a few more minutes for the rest of this morning. I want to just welcome you to the SSCI Presidential Symposium this morning. I'm excited about the science we're going to be describing later this morning, awards that we're going to be presenting, honoring young people for their efforts to date. So I think it's going to be a great session. This meeting and our SSCI organization, with a tradition now spanning 70 years that we celebrated last year, represent a celebration of academic medicine and multidisciplinary medical science, as well as fellowship and friendships that I can look around and see that span many decades. There's something unique about this organization and this meeting, the concept of bringing together trainees, investigators of all kinds, bench researchers, translational researchers, health services and beyond, clinician educators, and academic leaders from across medical specialties remains more relevant now than ever. The theme of this meeting, Harnessing the Immune System, is a topic filled with countless examples of the impact of translating fundamental science into clinical applications that enhance human health and result in innovative medical interventions. It is indeed a thing of beauty to watch scientific breakthroughs translated into innovations in healthcare and ultimately seeing the impact on our patients and their families. When I started my career in medicine, the diagnosis of acute lymphocytic leukemia in a child was frequently fatal, and the 5-year survival of young children with malignancies was just under 60%. When I started my career in medicine, there clearly has been progress since I started, but we have a long way to go. Progress has been made, there's significant room for improvement, and we're going to hear about some of those innovations that I think are going to take us to the next level in cancer treatment. The vaccine schedule was much easier to remember when I took my boards many years ago than it is today, but it was limited in scope, and our understanding of actually how those vaccines worked was very incomplete. We knew they worked, but we didn't really understand why. Advances in the fundamental knowledge of innate and adaptive immunity, T-cell function and inflammation have opened the door for exploiting that knowledge to more effectively prevent and treat human disease. Compelling examples include using the power of the immune response to treat malignancies in creative new ways, such as with checkpoint inhibitors, T-cell therapy utilizing chimeric antigen receptors, and therapeutic cancer vaccines, which collectively hold promise even with cancers that have eluded effective treatment for many years. Cancer immunotherapy was declared the breakthrough of the year by Science magazine in 2013 and was declared the advance of the year in both 2016 and 2017 by the American Society for Clinical Oncology. And it was listed as number 3 on the Cleveland Clinic's 2017 Top 10 Medical Innovations list. Dr. Sharma, who's joining us from MD Anderson this morning, will tell us about her work in cancer immunotherapy later in the Presidential Symposium. Other great examples include the custom design of improved vaccines against old and newly emerging microbial pathogens such as Ebola and Zika, and the power to selectively inhibit pathways of inflammation to reduce the ravages of autoimmune diseases and other chronic inflammatory diseases. Not to be overshadowed by cancer immunotherapy, vaccines were declared the #1 scientific advance of the 20th century for good reason. As you look at this slide, as you see the remarkable success in disease prevention over the course of the 20th century. And rapid development of epidemic battling vaccines using a very promising vesicular stomatitis virus vaccine platform was declared the #1 medical innovation of 2016 by the Cleveland Clinic. The new recombinant Ebola vaccine, in which a gene encoding the Ebola surface glycoprotein was inserted into an attenuated but replication-competent VSV vector, was found to be 100% effective with a single dose, and interestingly, using ring vaccination technique that harkens back to the success of the smallpox eradication efforts many, many decades ago. So new technology with old methodology in terms of the implementation. In a global effort that involved the WHO, government agencies from many countries, industry, foundations, and academic scientists, this promising vaccine emerged in record time. The vaccine was tested in almost 12,000 residents of Guinea within a year, a little over a year, of the onset of the deadly Ebola outbreak in West Africa. This is record time for vaccines that normally take 10 and 20 years for development. The VSV platform can now be utilized as a blueprint for future emerging pathogens, popping in the key ingredient, the key antigen, in a rapid response to an outbreak. Today, Dr. Rouphael will tell us about an innovative new approach to vaccine development that adds yet another new dimension to vaccinology by determining intricate molecular signals that occur in an effective immune response to antigen challenge using systems biology with the goal of optimizing vaccine-induced protective immunity. It's an amazing field, understanding the immune response and then using it to make a better immune response in terms of vaccine-induced immunity. These medical breakthroughs require a long journey from inspired hypotheses and key questions, plotting, detailed, and repetitive experimentation that many of our young investigators are being introduced to as we speak, the vision to see where fundamental knowledge can be translated into the realm of human health and disease, animal and human investigation, regulatory ethical considerations, understanding the epidemiology of the disease, understanding the practice of medicine and how it can be implemented in medicine and science. Beyond that, we need to educate our leaders, our patients, and our current and future clinicians on how to use these health innovations wisely. Advances in medicine result from partnerships and collaboration between fundamental scientists, translational scientists, funding agencies, regulatory bodies, and clinicians. Public and private partnerships can be a critical element in advancing the scientific discoveries. The nature of effective science has changed dramatically over the past century, and it has become a team sport. The physician investigator remains a key partner in recognizing how fundamental scientific breakthroughs can be applied and translated into medical innovations. We live in uncertain times with regard to federal funding for research. I believe we need to take every opportunity to emphasize the link between advances in fundamental science and the potential impact at the bedside and in the community. We must do what we can, everything we can, to maintain federal funding for basic, translational, epidemiologic, and implementation research. We need to do everything we can to maintain loan repayment programs and career development programs, collaborations with philanthropic organizations such as the Gates Foundation and others, collaborations with industry. And we need the robust support of professional organizations like SSCI and AFMR that celebrate multidisciplinary medical research and medical education to sustain progress and continue the amazing trajectory of medical innovations. I really am looking forward to our scientific presentations this morning and tomorrow morning, by the way, at the joint plenary session. This theme of harnessing the immune system will be continued. These sessions are going to provide detailed examples of how fundamental knowledge of the immune system is being harnessed to have direct impact on human health. So welcome to the SSCI Presidential Symposium, and thank you very much for being here. I appreciate the great turnout this morning, and look forward to having you enjoy the science that's coming ahead. So thank you very much. We're now going to have some fun with honoring some people in our organization. If I can get out of this. My cursor's not working. Can someone help me get out of this file and into the other? As we do that, I'm going to, so I need the business meeting slides up. But as we do that, the next thing on my agenda for this morning is something I'm very pleased to announce, and it is something near and dear to our organization. Mentoring, particularly mentoring trainees and young investigators, is really part of the mission. Got it? Yep, that's it. The mission of our organization, and we have created some awards that are highly competitive. I will say that there are many applications and nominations for these awards each year, and it's a difficult decision each year, but I'm really thrilled to announce our SSCI Mentor of the Year Awards this year. So our first one is someone who's being honored for his mentorship of postgraduate trainees, and that is Perry G. Rigby from the Louisiana State University Health Science Center. So let's congratulate. Our second award recipient is for his mentorship of junior faculty, and that is Richard McCallum from Texas Tech University Health Science Center. So congratulations. Come on up, Richard.
Thanks, Monica.
Thank you. This is really great. Very good. Congratulations. Well deserved. Okay, now some business. We're going to just go through the treasurer's report. I am acting for Galen Marshall, who is arriving later in the meeting from Hawaii, so I don't feel so sorry for him, but I'm going to act in his place for the Secretary-Treasurer's report. I think we can be brief. I think from a financial standpoint, we are in good shape. Our income exceeded our expenditures. Our fund for academic medicine is growing. We very much appreciate all of the wonderful contributions, and we hope they continue. Our journal finances are very healthy, and so our total balance is approaching $1 million. This is just the graphic way of looking at it. The green bar is our income versus expenditures in red, and our total balance. So healthy financially. Our membership is sustaining, I will say. We would love to grow it, and I will say that Mel Wilcox, who's taking over the presidency for SSCI for later this morning, is making it a priority for 2017 to build our membership. So we have 412 active members, we have 6 associate members, and 22 emeritus members. We are honoring 22 new members this year. So you can see we had, in the 1990s, we had, as the subspecialty meetings were growing, our membership went down, but I think you can clearly see that we are sustaining. We would like that trajectory to go back up in future years, and that's what we're looking for. So this is to honor our 22 new investigators and clinician educators who have been elected. This is an honor and an achievement to be elected into this society, and we really applaud the accomplishments. These are 22 people from approximately 12 institutions throughout the Southeast, and if you look closely enough, you'll see Oklahoma and beyond. So congratulations to each and every one of you, and we want to welcome you to a reception this afternoon at 6 o'clock at the Roosevelt Room, at the Blue Room at the Roosevelt Hotel, a few blocks away from here. So congratulations very much to our new members. This is a good year. We worried as the NBA All-Star Game forced us to change our schedule and get us reoriented to not having this meeting from Thursday to Saturday, which we've had for a century, it feels like. We, despite that, have seen pre-registration closed out near what our final registration was last year, and so this is not up to date yet. We expect something like 100 on-site registrations, so I think in general this is a good meeting, healthy meeting, despite the kind of the need to midstream change our dates. And abstract submissions, as you can see here in this bar, are up, so we're pleased. We had more posters than ever. We had to make people shrink their poster size so we could get enough of them into the rooms last night. And I think it was a really great event. Our Fund for Academic Medicine, which is something that Jesse Roman and a number of others in the organization have spearheaded, has continued to grow. We have over $600,000 in the fund. We are beginning to look to the future of how we can use these funds to benefit our young investigators and young members of this community. I love the fact these are people who have donated since its inception, and now we are spread over 2 slides, and we've had to shrink the font to fit everybody on here. We're happy to expand to 3 slides if we need to next year. So we very much appreciate your contributions to the organization, and we are going to be rolling out some, I think, exciting new opportunities in the future that will be a direct result of these contributions. So more to come later on that. We also have had contributors to the support of this meeting as listed here, and I want to point out there are some individuals who regularly support the meeting in addition to some corporate industry support for activities within the meeting. So thank you very much. Our website, I want to just point out we've gone through a major overhaul. We brought it into the 21st century. It's new, it's vibrant, there are lots of things on it. We want to get feedback from you. We want people to go there and use it. So please visit the website. There are lots of things you can go to that site and find more information about membership, about you can do your dues online, you can donate to FAME, you can access the newsletter and many, many more things that are within the scope of this website. So please use it, and I think you'll find it easy to use. And I want to, Vin Temprici and his committee on our council have done a great job of bringing this and updating our website. Okay, so now I'm going to transition and invite Dr. Ploth from Medical College or Medical University of South Carolina, who is our editor-in-chief for the American Journal of Medical Sciences, to give us our journal report.
Can you tell me how to get to my slide panel?
This is your slide panel. You just continue down with the down arrow, and you're there.
Great, fantastic. Madam President, ladies and gentlemen, I'm here to report very succinctly the journal is in great shape. That's not a biased editorial opinion either. However, it's been a year of movement. The office of the journal, located at the Medical University, has moved physically twice. And a lot of additional movement in that we moved to a new publisher, Elsevier. We had formerly been with Lippincott, LWW. We moved to Elsevier as of January 1, 2016. It's been a very favorable move. We have an attractive contract with them, and we have a wonderful working relationship. Things are going very well. And as you may see, I intended to bring one of the new journals with me that's under my seat, but if you've looked closely at the journal, you can see the format's changed a great deal, and I've heard a number of comments from members and others that it's much more attractive than it was before. So we hope we've improved it. Perhaps it can be improved further, and if you feel it can, please speak to us or speak to the new editor as he comes on board. One other movement was a change in our managing editor, and we have a new managing editor. Sarah, would you stand? Ms. Sarah McCann is our new managing editor in the office. And as our early managing editor departed, I thought our ship would sink. Sarah has not only kept the ship floating, but it's even come up higher in the water. So she's been a wonderful addition to our staff, and she's a joy to work with. Thank you. We also moved from an older editorial submission process, EVIS, back to our original editorial process submission, which is editorial manager. Name, quote, editorial manager. If you've looked at the website, you've noticed that. Please be aware of that. And although you may still be able to get into EVIS, if you submit a manuscript to us, please do not use it. Go back to editorial manager to submit your material. The state of AJMS is good. I'm not going to comment much except to say we need quality submissions. Please send us your best original research. A review, if you feel it's appropriate and timely, we would be delighted to have. And we need those. We tend to have a surplus of case reports, images, and ancillary reports, but primary research, investigator, hypothesis-originated research and reviews we need badly. One other statement that I would like to make is that I'm delighted to report to the membership that our impact factor substantially improved last year. We moved from about 1.35, I believe in the mid-50s in terms of journal rankings internationally, to a new impact factor of a 1.59, and that improved our status by about 10 places. So from mid-50s, we're down to the mid-40s in terms of international ranking for citations. In addition to those reports, in addition to those movements, we've had substantial improvement in our financial picture. It continues to be very positive. I'm not going to give you the nitty-gritty detail on submissions, but other than those comments, I would like to make one final comment, and that is there will be one more change in the editorial office this year, and that is I will have completed my term and there will be a new editor. And that new editor is proposed by council, and I urge you to vote for our single nominee, Dr. Jesse Roman. Jesse, are you in the room? There he is. So our new candidate editor, I assure you he'll do a wonderful job with the journal. It's been a pleasure to serve you. It's been a pleasure to be editor. Thank you very much for your support. It's been a wonderful run. Thank you.
This is a token. We actually have a very nice additional gift that is in preparation to honor the 10 years of service that Dr. Ploth has given to our organization and stewardship of this journal. You can see he's leaving on a high note, and we very much are very grateful and appreciative of everything he's done for this organization. So please give him another round of applause. A large framed picture of your first cover and the last cover on the way. So thank you, Mr. King.
Thanks.
Doing the Japanese
Very much.
Trump. Good. Thank you so much.
I'm Dutch.
Two kisses, he's Dutch. All right, I have a few announcements, and then we're really, and then we have one more award presentation, and we move on to the scientific portion. A couple announcements. Reminder that tomorrow, as part of your registration, so it's free lunch, there's a jazz luncheon. It's usually in this room. They reformat it into a lunchroom from 11:30 to 1 in this ballroom. So please come. It's good food and good music. At the registration desk, we are selling promotional materials, including ties and scarves and lapel pins that I'm wearing to demonstrate. So they're on sale at the registration desk. So now I'm going to call Dr. Ploth back. Oh no, I've skipped something, sorry. did skip something. I thought we were ahead of schedule, but we aren't. So now I'm going to ask both Bob Means and Tonette Cruzelwood up to the podium because we need to walk you through the nominations. Let's get you to those slides. The ballots, and she will walk you through that. And is he coming up to go through? I thought.
Yes, he is right after.
Okay, good. Thank you, Tonette.
Okay, great. Thank you. And good morning, everyone. Welcome to New Orleans. Hope you're having a wonderful time. The ballot should be on your seat, or you should have received one. We have several outstanding individuals who are on the ballot, a couple of running unopposed, but that's consistent with our practices. So President-elect Dr. David Guido from Emory, Editor-in-Chief of the American Journal of Medical Sciences, you've already heard, Dr. Jesse Roman from the University of Louisville. For our counselor, we need you to vote for two. We have three outstanding candidates, Dr. Lydia Bazzano from Tulane University, Dr. Paul Muttner from the University of Alabama, Dr. Ken Nugent from Texas Tech. And we have two nominees for our nominating committee. Please vote for one. The first is Dr. Carl or Chip Levy from Ochsner Clinic Foundation. Second is Dr. Cherry Wankertrall from Emory University. So it's very important that you submit your votes. We're excited about the quality of our candidates, and we're looking forward to moving the organization forward with new members. Now I will turn the next portion over to Dr. Means for the bylaws.
There you go.
Okay. So this year, you will receive with your ballot a ballot for the revision of the bylaws of the Society. The bylaws were last revised in, I believe, 2007, when the category of associate member was created. Heaven knows when they were revised before that. When we were reviewing the bylaws, it became clear the bylaws had been designed for a time when the major function of the Society was holding a meeting and all the members came to the meeting, so all business was done at the meeting. It did not allow, for example, opportunities to vote electronically like you do for most of the other societies. We also had a lot of committees, including standing committees that weren't listed in the bylaws. We had standing committees that were listed in the bylaws that didn't exist because they no longer had the function. We had processes that we hadn't been following, and so it was time for an extensive revision of the bylaws. You see on the screen before you an executive summary of the proposed revisions. They are primarily aimed at increasing the functionality and efficiency of the bylaws and reflecting the changes in the environment, like, for example, the ability to vote online. The committee structure has been revised to make fewer standing committees, which are primarily focused on operations, and other committees, which will still operate, will be created by the President and the Council as needed. It also establishes terms and provides some opportunities for election of candidates, of committee members from the larger membership. So in any event, one of the features of the current bylaws is that we have to get paper ballots. In order to change the bylaws, we need paper ballots at the meeting, and the election has to involve 20% of the active members, which means we need 85 ballots. When I walked through the door this morning, we had 50 in hand. Fill out your ballots and turn them in to Joan and Pam, and we will get them and we'll get them done. Otherwise, I will be pursuing you at the reception tonight and possibly into your future. So again, thank you. In the tradition of New Orleans, vote early and often. Thank you.
Thanks, Bob. And thank you for all the hard work. It's tedious work to revise bylaws, and Bob and his group did a great job of it, so thank you very much. So now I'm inviting Dr. Ploth back up to the podium to tell us about the Tinsley Harrison Award and make the presentation.
I believe you all know that Dr. Tinsley Harrison at the time of his retirement and death, was on the faculty long time at the University of Alabama, Birmingham, although he had been on the faculty of several other southeastern schools through the course of his career. A personal vignette is I started as a very junior faculty member at UAB, and my office was almost next door to Dr. Harrison's. And one of the most embarrassing admissions I have to make in my life is that I was so intimidated by him that I did little more than say hello to him in the hall. And I would like to go back and beat myself up for that, because I would have loved to have gone in and just introduced myself and gotten to know him better than I did. But nonetheless, he was the founder of this society in 1946. And in honor of his huge contributions to both this society and medicine, the American Journal of Medical Sciences has named an award for him. And that award is designated to what the editor and associated editors, 4 of us, select as the best manuscript published in that calendar year. And the winner this year is Dr. Ihab Hajar from Emory University, an internist. Dr. Hajar, come up. The title of his paper is Circulating Progenitor Cells Linked to Lower Cognitive Decline in Healthy Individuals. And it was published in the American Journal of Medical Sciences. Luke Verillo from Elsevier is going to present Dr. Hajar with his award. Please come up to the podium, if you will.
Thank you. Congratulations.
You should get that.
Yes, and then we'll get Luke. Got it. Congratulations.
Thank you.
The 3 of us. Good. All right. Thank you. Congratulations. Thanks, man.
We may need help getting the, all right. So now we're going to proceed to the scientific presentations for the Presidential Symposium. And our first speaker, it's my great pleasure to introduce Dr. Nadine Rouphael, who is from Emory University, where she is the director of the Emory Vaccine Center, Emory Vaccine Treatment and Evaluation Unit, that is, the VTEU, at the Hope Clinic at Emory. This is the clinical research arm of the Emory Vaccine Center. She's also the director of the Human Immunology Project Consortium at the Hope Clinic and the national lead on many VTEU projects. She's an associate professor in the Division of Infectious Diseases, and it's been my personal honor, and it's so gratifying to have watched her career develop and her success story materialize. She's been doing wonderful work. She's going to be talking to us today about her work with Bali Palindrom from the Emory Vaccine Center using systems biology that I mentioned earlier. These approaches are used to better understand and exploit the immune response to better our vaccines and to better our understanding of how we respond to antigens in general. So it's really my pleasure to introduce Nadine and welcome her to the stage.
Good morning, everyone. I am really honored to be here and would like to thank Dr. Monica Farley and the organizer for inviting me. I'm an infectious disease physician by training. I'm a researcher by passion, and I do work with a talented team of scientists and staff at the Hope Clinic, and that's the clinical arm of the Emory Vaccine Center. Our mission is to address global public health threats by translating basic research discovery into clinical advances. And we aim to achieve our mission by partnering with education, engagement, and participation of the community, training the next generation of investigators, and performing clinical trials of vaccines and treatment, but also conducting translation human immunology studies, which most of my talk today will focus on. We are sponsored by many federal and state agencies, mostly through NIH, and this brings me to my conflict of interest. So the objectives of my talk today is to introduce you to the concept of system vaccinology, guide you through different examples using different vaccines and how system biology can be applied to them, and how as a result that had led us to have new immunologic insights. How can we apply system vaccinology in different populations, special populations that need more tailored vaccine to them? And how one example to make the immune response better is the use of adjuvant, and how can we dissect that from an immunologic perspective. So variation in genes and environment contributes to producing a wide range of individual phenotype, a humanity that's so rich and diverse. But can this diversity be captured? The last decade confirms that it's not only possible, but it can be done with great precision. This is the power of big data or system biological approach, where all elements of a system that can be measured will be measured. System biology of vaccine, one of public health's greatest achievements, can hopefully allow us for the first time a personalized preventive medicine. And this is the time to modernize in medicine. Other aspects of medicine did. So this is how Atlanta patients were taken to Grady last century. And this is recently how our Ebola patient arrived to Emory hospitals. And we modernized vaccine by moving from the trial and error efforts of the Pasteur era, moving away from isolate, attenuate, vaccinate, to create a rational design of vaccines that can be safe and effective in diverse populations, such as the extreme of age or with weakened immune responses, and for diseases where effective vaccines are still desperately needed, such as tuberculosis and HIV, for example. So even though vaccines have been around for many centuries, we really don't know exactly how they work. And unless we do that, we cannot design appropriately the next generations of vaccines. In system biology, we probe the immune system by causing a synchronized perturbation using the vaccine as a tool. This happens few seconds, few minutes, few hours, few days after we inject the vaccine. This is the so-called day zero that I will refer to a lot during my presentation, and will be followed by an intense phlebotomy schedule to allow us to better understand all the mechanisms that are driving the immune responses. Typically, the correlates of protection of vaccines are largely based on antibody response. And very little is actually known about this black box in immunology called innate immunity and the different metabolites that are secreted in the body very early on after immunization. So classic teaching shows us that the innate immunity is not specific, has no memory, and is very much useless unless it's followed by adaptive immunity. But the thinking here is like actually innate immunity is the one that will be shaping the downstream adaptive immunity, and how can we understand it better to design better vaccines? So to unlock the mysteries of the immune system, this can be done through system biology by measuring the number and the function of the different immune cells, the multiple cytokines level, the type of genes that are upregulated or downregulated, and other protein, lipids, sugars that will help us identify, with the help of very expert biostats, the vaccine signatures that will correlate with protection. How did we apply system biology to understand successful vaccines? So this will allow us to build a platform to test future vaccines. The numbers of publication on system biology and vaccine is steadily increasing over the past decades. And then Bali Pulendran from Emory University is credited to be one of the pioneers in the field. The first studies from his labs focused on yellow fever and influenza vaccines recipient, and showed that a BAF, or a B-cell activation factor gene expression, highly correlated with the predicted antibody response to those two vaccines. So that could be possibly suggesting the presence of universal vaccine signature. And if indeed this is true, that will be the holy grail of vaccinology. So imagine this. We have a universal vaccination signature that can quickly screen many potential vaccine candidates and cut down drastically on cost and lengths of R&D. So to find out if this is true, we needed to test more vaccine. So we compared data from the trials of influenza vaccines and the yellow fever vaccines to a trial that was funded by the Emory University Research Committee, where we enrolled at the Hope Clinic 30 participants to receive one of two vaccines. One was the MCV4, which is a protein conjugated meningococcal vaccine, and another one was the MPSV4, which is a polysaccharide meningococcal vaccine that contained 4 meningococcal serogroup A, C, Y, and W135. And we looked at the immune response. We looked classically at the antibody response, either from a quality perspective by doing serum bactericidal activity assays or by measuring the quantity of antibody, and that's by ELISA. We also looked at the antibody secreting cells. We looked at the different types of cytokines. And most importantly, we also looked at the gene expression in the peripheral blood mononuclear cells. That's PBMC. That's a collection of lymphocytes, monocytes, and macrophages. And when we study the gene expression after vaccination, that could be tricky because we're looking at thousands of genes and we're looking at changes that could be really small in the PBMC profile. So that makes it very difficult to be able to distinguish a signal from the noise. In red, you see the genes that are upregulated, and in blue, the genes are downregulated. And in the case of the meningococcal vaccine, the first two here, where we use a conventional gene-by-gene analysis, we had a very disappointing small number of differentially expressed genes. So we decided to analyze the data a little bit differently, and I will walk you through what we did. So in a typical experiment, expression of thousands of genes, we call them here G1, G2, G3, are measured in many samples. We're going to call them S1, S2, S3. And the correlation of expression level of each gene with all other genes is tested in a linear and nonlinear association. So networks are actually modeled by connecting the genes, those are called nodes, with lines, those are representing edges. So actually, the interaction network is then compiled of all the nodes and edges, and the model in particular are identified as densely connected area of a network. We then merge 32,000 PBMC gene expression profile from 540 published studies. We identify 334 different blood transcriptome models. So when those models were combined with a robust statistical framework, this revealed 3 distinguished transcriptome program in vaccine antibody response. So you see here the protein recall response that correlated with the antibody response to the influenza vaccine, and also the antibody response to the DT portion of the conjugate vaccine. A primary viral response, very typical of a live attenuated viral vaccine, such as the yellow fever vaccine, and an anti-polysaccharide signature shared by the response of the polysaccharides portion of both the polysaccharide and the conjugate meningococcal vaccine. So all those interactions can be visualized with the familiar hairball that system biology absolutely love, but probably not all of us here in the crowd. This might exacerbate some people in the audience, probably including me, but they do really represent a fair approximation of how the cell business is transacted. It's much more robust than the simple reduction association that we have been accustomed to. However, we still need to better understand the biology of each and every transcriptome model to understand how vaccines work and apply them clinically. And I will just give one example of how we did that with one of the immunologic insights resulting from the flu trial. So when we conducted the flu immunization study, there was an unexpected finding where the antibody level at day 30 after vaccination correlated with the expression of TLR5 3 days after the flu shot was given. This is very interesting because the natural ligand of TLR5 is flagellin. and flagellin is found in many bacteria in the gut. So is it possible that our microbiota can actually help boost our immune response to vaccine? To actually test this hypothesis, the flu shot was given to TLR5 deficient mice, as well as mice treated with antibiotics and germ-free mice. And we measured their immune response to wild-type mice receiving the flu shot. And the result was really striking, with a significant decrease in the immune response in the mice that had less bacteria in the gut, suggesting an important role of the microbiome in vaccine immune response, not only for the flu shot, but we were able to find that it was similar for an inactivated polio vaccine as well. So some clinical implication of that could be whether a patient should be vaccinated with flu shot at hospital discharge, when typically 40% of people during their hospital stay receive antibiotics. So we are currently conducting a study right now to better understand the effect of antibiotic on the immune response to influenza vaccine. And then more practically, how can we help a population that typically doesn't respond so well to vaccine respond better to vaccine? And one example is in the elderly. The elderly is the fastest growing segment in our population, and worldwide it is expected that by 2030 we will have more than 1 billion seniors. Application of system biology to vaccination in the elderly may help to identify gene signature that will predict the suboptimal responses and the identification of specific defects that may be used to target novel vaccination strategy that will improve efficacy in elderly. And those are few examples of what the defects in vaccine response in elderly are based on the literature. There are vulnerabilities at many levels, from the innate immunity with the dendritic cells, but also to T cells and B cells response. So since 2010, Emory had been awarded a U19 grant by the Human Immunology Project Consortium, or HIPSI, from NIH. So within HIPSI, we have conducted many trials comparing young versus elderly, and they're using a system biology approach and applied to different vaccines. One of them is influenza vaccine, the other one is the live attenuated shingle vaccine, and the pneumococcal conjugate and polysaccharide vaccine. My focus today will be mostly on the influenza vaccination in the elderly. As probably all of you know, the toll associated with influenza morbidity and mortality in the US is high. The best way to be protected against flu is vaccination, and despite more than 65% coverage in elderly population, flu is still a huge problem for our seniors. Older individuals, and especially those that suffer from chronic medical condition or immunologic disorder, account for 90% of all influenza-related death. Unfortunately, the same immune defects that render them susceptible to infectious disease will also prevent them from developing a protective immunity following immunization. We conducted a simple trial where we stratified subjects by age. Younger were those between 25 and 40 years of age. Elderly were 65 and above. We gave everyone the flu shot at day 0, followed by an intense phlebotomy schedule at day 1, day 3, day 7. And we also measured their antibody response to influenza 4 weeks after vaccination and then 6 months after vaccination. And you can see first with the results of the antibody for the subject, you can see clearly that the Y, which is the young group, compared to the E, had a higher antibody response measured by hemagglutination inhibition assay. You can also see that there was also probably a log difference between their plasmablast response, those very important cells that actually produce the antibody, between the younger group versus the older group. And that was also seen in the Ig isotype, mostly for IgG, but also for IgM, which almost no response in elderly. Interestingly, we found that innate immunity cells, such as NK cells, were higher in elderly compared to young, and this also applies for the monocytes. But interestingly, you also see that the elderly at baseline, even before vaccination, had a higher level of innate immunity activation. We found many gene signature, and by far the largest difference we found between the young group and the older group was on day 1, where the young exhibited a much greater number of both upregulated, seen here in red, and downregulated in blue, compared to the elderly. We also found clusters of highly correlated genes among the TIV-regulated genes, and that's in young, we call them Y1 to Y6, and in elderly, and we call them E1 to E5. Genes in common to cluster Y4 and E3 were actually associated with antibody-secreting cells genes, and the overlap between Y1 and E1 contains several interferon-related genes. And here you can see how this actually is showing. And you can see actually the kinetic looks pretty similar between the two groups. But however, the magnitude of the expression of those interferon-related genes seems to differ drastically between the young and the older cohort. This is a slide that's done by age. And you can see in red, again, activated, in blue, downregulated. And you can see in the older, we see much more red for the innate immunity cells and function. That's the NK cells over here, the chemokine, but also the myeloid cells, and the regulation of the antigen-presenting cells, and a downregulation of the so-needed B cells, plasma cells, and immunoglobulin. And then we wanted to look actually at the duration of protection, which is very important. And then we found that originally a lot of people actually responded to vaccination, whether they were young or old. But 6 months later, we only had half of people responding and the other half not responding. And we measured that by checking in the blood if the antibody level was above 1/240. And then that's really interesting. We were able to find several models that were associated with cell movements and adhesion. And if those were present, which could suggest the successful migration of immune cells to the site of injection and inflammation, post-vaccination, that can predict that longevity response. So one of the gene is called P-selectin and plays an important role in the adhesion and the extravasation of leukocytes to the site of inflammation. So it's really nice to have a gene so early on at 3 days that can tell me 6 months later who's going to be the responder versus the non-responder. So now that I told you about what we have done in adults and elderly, I will focus right now within the HIPSI is more to move forward with better understanding the children's and the infant immune response, this other extreme of age that typically doesn't respond so well to vaccine. And how can we improve that? We can do it by adding an adjuvant. You might say, well, what is the adjuvant? So this is a particle that will boost the immune response. And actually, we use adjuvant in almost all of the vaccines that are available in the US, and it's alum. But the problem with alum, it's rather a weak adjuvant. So another one that's being used is MF59. It has been given to more than 80 million people worldwide, and it's licensed in many countries, including the US since last year, and it's given to elderly 65 and above with the seasonal influenza vaccine. So how does the MF59 really work? It does enhance antigen uptake by dendritic cells and the transport of that antigen to the lymph node. It also increases production of chemokine that are responsible of that initial inflammatory response at the site of administration of vaccine, and promotes the differentiation of monocytes to dendritic cells, and overall lead to an increased antibody production. MF59, as well as other adjuvant, have been heavily used in pandemic influenza. Typically, we don't respond to an influenza strain that our body had not seen. One of the examples here shown is H5N1 with or without MF59. So you can see that if you didn't have the MF59, you really had a very poor response that was less than 10, so not significant. But if actually the adjuvant was used, participants after 2 doses of H5N1 with MF59 were able to mount a nice response. And this is independently of how much antigen you give. 15 micrograms is a typical dose that's found in a flu shot, but we can go even down to 375 microgram with MF59 without affecting the antibody response. So obviously, one way to apply that is in kids. So this study had actually been done in Europe with balipulindrin and claircycrese. And they vaccinated children between the age of 14 months to 24 months with the flu shot, with or without MF59. And then TIV will refer to the trivalent inactivated influenza vaccine, and ATV will be the adjuvanted trivalent inactivated vaccine. And I think it goes without saying that when the adjuvant is used, you can see a better immune response, at least for H1 and H3 strains, and overall for the 3 strains. We also looked at the multi-cytokine production by the vaccine-specific CD4 cells, and you can see particularly for a TNF alpha and IL-2 expressing cells, this difference was drastic, as well as the TNF alpha, interferon gamma, and IL-2 vaccine-specific CD4 T cells. This is a busy slide. It actually showed the number of differentially expressed genes, and I want you to focus on the D1 and the D3 from the regular influenza vaccine, and then you can see that the number of differentially expressed genes here is much less than those that are expressed D1 and D3 with the adjuvanted vaccine. Nonetheless, when we compare the infants and the younger children, differentially expressed genes, they are still much lower than the adult ones. From our historical cohort. So we tested in elderly, we tested in kids. We've also using a more comprehensive approach to system biology. This is just an example of the unpublished data of the shingles live attenuated vaccine. So here our focus was not only gene expression, cell population, and cytokine, but also we wanted to know what is the amount of metabolites that are being secreted early on. And this is, to our knowledge, the first study that will integrate those different platforms to a vaccine. So in summary, system biology through different platforms related to genomics, but also lipidomics, metabolomics, glyconomics, powered by a robust data modeling and integration, will allow us to better understand the immune responses to vaccine, reprogram that suboptimal responses in the extreme of age and in immunocompromised patients to design the safest, most effective vaccine moving forward to those who will need them the most. And that can help understand health in general in a more individual manner. This is, again, the work of many collaborators, colleagues, and friends. I would like to thank very much balipulindrin, who I met in April 2008, and my academic career had changed since. Rafi Ahmad, the co-PI on the Hep C studies, all postdoc from Mario Cortese, Dimitri Kazmin, Nakaya Helder, and many other at the Hope Clinic, my mentor, Mark Mulligan, Mary Bauer, who had conducted all the studies in elderly. She had enrolled more than 250 subjects and had vaccinated, had given more than 300 doses to make the system biology with the studies in elderly possible. Alison Beck, also, who's our project manager at the Hope Clinic. Evan Anderson, he's our new collaborator within the Hep C, since we're trying to understand better how the immune response in kids will be. Our upcoming study will be to understand, using a system biology approach, the response to chickenpox vaccine as a priming dose given between 12 to 15 months compared to the boost dose given at 4 to 6 years of age. At Emory, David Stevens, Monica Farley, Anish Mehta, Abir Moana, who had served as the site PI at the VA for the pneumococcal vaccine study, Tianwei Yu, our statistician, my mentors at the CDC, and our collaborator in Denver on the shingles vaccine. Thank you so very much.
Thank you, Nadine. Can you stay up? And, we can take questions. I think we have time for a few questions. I can start it out as people, he's bringing a microphone to the center. Can you tell me about what you're learning of the differences in the response or the adjuvant efficacy with regard to the patterns? Can you predict based on, and are there major differences between alum and the newer oil and water kind of adjuvants in terms of the signature?
Yeah, definitely. There is a big difference between the adjuvant. I didn't talk about another one that was actually even, we think the signal is even more amplified with the ASO3 one compared to MF59. And actually what we see is not only a much more increase in the upregulated and downregulated genes at day 1, but we also see a shift. So whatever for the unadjuvanted vaccine, we'll see that response, but at a much lower scale and much later. For example, at day 3 compared to the adjuvanted one.
Jim.
Hi, Jim Oates, Medical University of South Carolina. So for the younger folks in the room, why go to a multi-specialty meeting? I'm very excited because the patients that I see auto-vaccinate when they go out into the sun and have necrotic keratinocytes, and they express the same genes you're trying to induce, and they have lupus. And one of our target therapies is to interfere with the type 1 interferon pathway. So how is this going to affect vaccination? How do we target those pathways? This is actually very useful information. Are you aware of folks like yourself in inserting yourselves into these trials of interferon receptor antibodies and interferon alpha?
Yeah, I mean, we would be very interested. We had looked particularly in our second round of Hep C to target more like transplant and better understand. But, as you know, it's a wild population. The variation between the immunosuppressed regimen that are used is very wild. And actually, we actually considered looking at the lupus patient. I know in the HIV vaccine world, they're really looking at the lupus population in a way to target the immune regulation, which will help them understand better what type of immune response to expect when this is happening.
Yeah,
Thank you.
Sorry, on that.
Gary Gilkson, MUSC as well. Do you notice any sex differences in responses?
We know that there are sex differences in responses. I didn't detail that much. There are other groups that had looked at it more closely with actually difference not only male to female, but also the country where the parents were born, what's the ethnicity, the race, all that can affect that. I think we will do much more like analysis on that moving forward.
One other question, if you get multiple vaccines at the same time, like it happens when you go in the military, you go overseas, does that enhance responses or inhibit, or does it not really have any effect?
Right, we haven't studied it from a system biology approach. We think we already have a lot of noise that exists, but there is data just looking from an antibody response that this is still okay, from like a very crude way that it's still actually fine to proceed this way.
Thank you.
Excuse me, I have a brief question.
Yes.
I'm a cardiologist. I just want to know, are there any medications that could enhance the effect of vaccination?
Repeat the question.
So the question is like, is there any medication that can enhance the vaccine? I mean, by medication, I think more about any type of molecules that could serve like an adjuvant-like. Will be it, and then the big example will be ASO3, MF59, etc., and avoiding medication that can actually suppress the immune system, such as steroids or other inhibitory molecules.
Is there any evidence that you could, at this point, customize, and if you knew the country of origin of their ethnicity or their gender, race, etc., is there work being done to say, okay, this adjuvant would work better in this demographic group, or sort of customizing, personalizing the choice of vaccines for the future?
Right. I mean, that's the way we're headed with the additional studies. And then actually, particularly the Hep C is engaging more in Europe and in Africa, such as Gambia side, to be able to understand that difference that's related to the environment, what they eat, the type of microbiota that they have, but also the genes that are specific to the different population around the globe. Yeah.
All right. Any other questions? If not, thank you so much for a great presentation. That's great. I'm excited to move on to our next speaker. Dr. Padmini or Pam Sharma from MD Anderson is joining us. She's an MD PhD who is a professor of genitourinary medical oncology and immunology in the Division of Cancer Medicine at MD Anderson at the University of Texas. She's a well-funded investigator, has a wealth of experience in the area of immunotherapy for cancer, and is going to talk to us today about the use of checkpoint inhibitors in her work, both from the lab to the bedside and back. I do think we need, for some reason, we don't, the cursor, oh no, now it is. It wasn't working, the cursor wasn't working before, but now we're good. Okay, so welcome, Dr. Sharma. Thank you for being here.
Thank you. So thank you to the organizers for inviting me. I'm very happy to be here today to present our work dealing with immune checkpoint therapy, and it's certainly been an exciting time, as you've heard, in cancer immunotherapy. The field has moved rapidly. I actually think the clinic is outstripping the basic science understanding, so it's really very important for us to try and get back to the translational research to help us with some of the understanding that we're seeing in some of the objective responses we're seeing in the clinic, so we have a better understanding. So just to make sure I show my disclosures here. As you are all, or mostly aware, T-cell responses really are the major soldiers of what we consider to be the responses we're looking for in cancer immunotherapy studies. And I think for years we were very focused on how to drive these T-cell responses based on our understanding of the T-cell receptor, knowing fully well that the T-cell receptor needed to engage with the major histocompatibility complex and antigen. So this was signal 1, and we all designed multiple vaccines based on signal 1. And so I'm sure you all remember years and years and years of vaccine study, just thinking that if we could just engage the T-cell receptor, we'd have a great immune response. However, that did not work out so well. There were hundreds of clinical trials done with antigens and vaccines targeting just specific, what we thought were tumor antigens at the time, and unfortunately did not lead to the type of objective responses that we wanted to see in our patients. And I think the field then moved forward to help us understand that the T-cell receptor is not the only signal that's important, that you needed co-stimulation as well, with CD28 binding to B7. And actually it's these two signals together that then drives a T-cell response. And again, that changed what we did in the clinic. A lot of then we moved to dendritic cell vaccines because we thought the dendritic cells then provided the co-stimulatory signals, and so we could have a better chance of driving an immune response that way. Unfortunately, again, we did not see a lot of significant clinical responses despite the two-signal targeting that we tried with our vaccine strategies. And this really had to do with further understanding that was needed. So it was really the basic immunology or the basic research that led to this understanding that T cells, although they require two signals to be turned on, the on signal actually drives an off response. So it's really the yin and the yang of the immune system that you're going to see throughout the presentation. It's that the immune system is driven to do its job, but it also regulates itself very tightly so as to make sure that it does not continue for prolonged periods of time. And so this identification of CTLA-4 as a third signal or as the inhibitory signal that turns T cells off was very important for then driving the field forward further. And of course, this was done by Jim Allison's group. And based on this understanding of CTLA-4 as an inhibitory pathway, Jim actually proposed that as tumor cells are dying and then presented by antigen-presenting cells, you could turn on an immune response, but that would also lead to CTLA-4 expression, which would then turn off the immune response. And in the setting of large, bulky tumors, then it would not give the T cells long enough time to work. And so Jim proposed actually that you could block CTLA-4 with an antibody, and by removing the inhibitory pathway, you can then allow the T cells to work for longer periods of time. So I know that the title of the meeting is Harnessing the Immune Response. Actually, we think of it as unleashing the immune response, because you're actually blocking the inhibitory pathway and allowing the T cell to do its normal job. And so in the first phase 1 clinical trial that we did, and for those of you who do phase 1 clinical trials, you understand that this is mostly a safety study. So for us to see clinical responses that are so dramatic, very rare in most of our phase 1 studies. But in this phase 1 study, we actually had patients with metastatic melanoma who had metastatic disease to brain, to lung, to liver, who had failed multiple other therapies. And then after 1 dose of the anti-CTLA-4 antibody, metastatic disease, as you can see here on the CAT scan, including metastatic disease in the brain, completely regressed and went away. And we had 3 responders like this who are still alive today. So this led to, of course, a phase 3 clinical trial with anti-CTLA-4. There were long-term durable responses of about 20% of patients, and that's really key because, again, most of us in oncology are used to the fact that we would increase median survival by a couple of weeks or a couple of months, but all the patients still die, and that's a real problem. But here we were seeing for the first time 20% of our patients who are alive 10 years or more. And again, these were patients who would normally die within a year of metastatic disease. And so this was amazing for us. This led to FDA approval of anti-CTLA-4, now known as ipilimumab, in 2011. However, anti-CTLA-4 is just the tip of the iceberg. It actually opened an entire new field called immune checkpoint therapy. And if you look here at this cartoon, you can see that there are multiple pathways that can be targeted and are important in the immune response. CTLA-4 was just the beginning. The PD-1, PD-L1 pathway is the next pathway that has clinically been targeted and led to FDA approval. And here we see that, again, in this phase 1 clinical trial with anti-PD-1, we could see a patient with metastatic renal cell carcinoma, and again, in the top panel, you can see the regression of a big bulky nodule. I want to point out the middle panel. This is a bone metastasis, and for those of us who treat patients with metastatic cancers, we know that bone metastases do not respond to most treatment. And here you can see with this immunotherapy, this bone metastases completely resolves. And so again, when we took biopsies from this patient, we could see that not only was the lesion disappearing, but infiltrating T cells were now accumulating within the lesion. Similarly, anti-PD-1 here is treating patients with bladder cancer, and each line represents a patient. The lines decreasing in size shows tumors that are regressing, and you can see some patients where all tumors are regressing, and the lines increasing some patients who are not responding to therapy and their disease is progressing. What I want to point out is that despite the tumor type, melanoma, kidney cancer, lung cancer, bladder cancer, despite the tumor type, it's the same therapy that's working. And because it's working on the immune system, we're not targeting the cancer, we're targeting the immune system. And this is a very important concept for us now because we've gotten to the era of genomic medicine where we were sequencing everyone's tumors to try and understand what to treat the tumor with. And actually what we're learning is that if you can actually engage the immune system is there to recognize the things that do not belong there. And so actually any tumor type doesn't belong there. And the fact that the tumor requires mutations in order to grow, those are the mutations that allow for the antigens to be presented so that the immune system can actually eliminate the cancer cells. So now this is where we are between 2011 and 2017. This is the FDA approvals that have all occurred as a result of immune checkpoint therapy as a development of the new field. And so this has been a huge paradigm shift for those of us in oncology. You can see we have now new treatments in melanoma, lung cancer, kidney cancer, Hodgkin's lymphoma, head and neck cancer, and bladder cancer, and even more to come as the clinical trials are ongoing. So it's really a revolutionary time. It's really an impressive time for us to be seeing our patients year after year after year after year, and they're still doing fine. There are issues that need to be addressed clearly. What are the cellular molecular mechanisms involved in these anti-tumor effects? As I said, I think the clinic is outstripping the basic science, and so we have to get back to understanding some of the mechanisms. There are also toxicities involved because these agents are not like typical chemotherapy, so they have different toxicities associated with them. And I'm not going to have time today to talk about the toxicities, but it's really a matter of education of how to recognize what they are, since they are different from other therapies. Can we identify predictive, prognostic, and pharmacodynamic biomarkers, what are the best standard of care therapies to combine with immune checkpoint blockade, and how do we combine chemotherapy with these agents, or how do we combine radiation therapy with these agents? Can we identify new targets to help improve the numbers of patients who benefit? Clearly, 20% is a big number, but we'd like it to be even higher than that. And then why do some patients respond and others do not is actually the big overarching question that we're constantly asking. So to do this, we actually set up what's known as the immunotherapy platform at MD Anderson, and it moves very quickly between cancer immunotherapy clinical trials and laboratory interrogation. As of September 2016, these numbers are a little outdated, but we have over 100 ongoing clinical trials across 15 different departments. There are 2,165 patients enrolled to date, 32,000 blood samples, 1,700 fresh tumor samples, and about greater than 22,000 archival samples that have been studied in order for us to understand how these immune checkpoint therapy agents are working. For us to do this, we really have to understand how to integrate laboratory and clinical research. And so this slide is very good for us to try and put these things into context. From a basic science standpoint, we know that hypothesis testing is most efficient when one variable is tested at a time. So in mice, we have an inbred setting, and the disease is homogeneous. If I want to understand what a particular gene does, I knock it in or I knock it out. In my patients, which are polymorphic and the disease is heterogeneous, my IRB is not letting me knock in or knock out anything yet. So the most I'm going to do is conduct these clinical trials, and I'm going to get some hypotheses that are generated from them. And so as a result of the hypothesis generating, though, it really is important to then connect back the dots to then hypothesis test, because this is the only way to truly understand the mechanisms of what's ongoing. The other thing we had to do was actually rethink how clinical trials are done. Again, most of you may be familiar with the phase 1, phase 2, and phase 3 setting, where phase 1 is safety and dose escalation, phase 2 is efficacy, and phase 3 is comparison to standard of care, which these are very important pieces. However, we've now proposed that we need the pre-surgical or tissue-based trials, which are we call the phase 1A or phase 2A trials, and these are done in parallel with our phase 1, 2, and 3 trials. And really, these are all about biomarker analyses and mechanistic insights. It's all about collecting the samples from these patients in order for us to ask the right questions. So the first trial we did this way was this pre-surgical clinical trial. This was our first phase 1A, phase 2A trial, if you want. And really, if you look, you can see this was 12 patients. So most of these trials are 10 to 20 patients. They're not large numbers of patients. They're done basically to ask a particular question in that cohort so that we can understand the mechanisms a little bit better. These are not large clinical endpoints, survival, response rates, those kinds of things. This is really mechanistic studies. So in this cohort of 12 patients who were now scheduled to undergo surgery because of localized bladder cancer, we asked them to participate in the clinical trial, where we can then give them the anti-CTLA-4 antibody. The patients are all scheduled for surgery, so we have access to the entire surgical sample after they receive 2 doses of the antibody. We have a cohort of stage-matched patients who go directly to surgery because that's standard of care, but they do not receive the antibody, and that's the control cohort. We're now able to study what does anti-CTLA-4 do in the tumor microenvironment, so as to better understand some of the mechanisms. And again, I can't stress enough the importance of having sufficient tumor tissues to do these types of studies. Most of the time, we're used tiny biopsies, and that doesn't give you enough cells, for, especially to study the immune system when it's made up of so many complex different cells. So here we have access to the entire bladder in this setting. Our pathologists are very key because they help us to make sure that we get access to the actual tumor tissues and identify which samples have tumor and which samples don't. And then we can use the tumors for multiple different assays as shown here. So one of the first things we did was we can look from IHC analysis at the pre-therapy and post-therapy samples. And it's very obvious, actually, when you're looking at these samples, the difference between the pre and the post-therapy. It's just large numbers of infiltrating immune cells, without a doubt. That's what immune checkpoint therapy does. It just drives an immune infiltration into those tumor tissues. And then when you stain, you can see that these are CD3 T cells, CD4 and CD8 T cells. There's some granzyme indicating activated T cells. Very few B cells, actually. So CD20 is a stain for B cells, and we don't see a lot of the B cells. So it's really a T cell predominant change within the tumor tissues. Now that we have this T-cell predominant change, when we looked at the differentially expressed genes between the pre- and post-treatment tumor tissues, it's not surprising that the top 10 pathways are related to T-cell pathways. And so you can see here, ICOS, CD28, T-cell receptor signaling, all of these are related to the fact that we now have increased immune infiltration in the post-therapy tissues. What was surprising was to find the ICOS pathway at the top of the list. So ICOS actually stands for inducible co-stimulator. It belongs to the CD28 CTLA-4 family, as you see here. Its expression has been known to be increased on activated T cells. However, in the literature, it has a very diverse role. It has a role in regulatory T cells, which suppress immune responses. It also has a role in TB cell collaboration in the follicular helper T cells of lymph nodes. A role in anti-tumor responses had not been shown or established prior to our work, and so it led us to ask multiple questions about why ICOS was the top gene and the top pathway we were seeing. So we began, because we had access to all of the tumor tissues, we could go back and look at the tumor tissues to see if the gene expression data was actually correct in terms of flow cytometry data and expression of ICOS on T cells. And here you can see in non-malignant tissues, there's about 13% of ICOS positive CD4 T cells. And in untreated tumor tissues, this is about the same at about 16%. However, after treatment with anti-CTLA-4, there was a significant increase, and we saw this in multiple patients where it ran up about 2 to 3-fold. And again, we had the access to the tumor tissues. So here I'm showing you the tumor tissues that have staining with H&E and the CD4 T cells. Some of these tumors also expressed tumor antigens, such as the NY-ESO-1 tumor antigen. And so for us to understand the function of these ICOS positive T cells, we could then sort the ICOS positive T cells from the tumor tissues and then use the tumor antigen in an in vitro assay to ask whether or not these T cells were capable of recognizing these specific tumor antigens. And here you can see that these ICOS positive T cells actually produce interferon gamma, TNF alpha, and MIP1 beta, which are all effector T cell responses against the tumor antigen. So although the literature was diverse about the role of ICOS positive T cells, in this particular setting we were showing that the ICOS positive T cells were actually effector cells. And we had a lot of skepticism when we first published the data, because again, ICOS is only known to be in the lymph node setting. That's the only place we see these ICOS positive T cells. So this was a different setting to show that actually they could play a role in anti-tumor responses. Because we now had access to data from tumor tissues, we can go back and look in the peripheral blood to see if this was also true. And I will say most of the time, a lot of our immune monitoring data, everyone's looking in the peripheral blood because we have access to peripheral blood. It's easy to look in peripheral blood. It's actually somebody who told me when you lose your keys, you look under the lamppost for the keys, but it could actually be somewhere that there's no light shining. And I think that was the issue. We were always looking in peripheral blood, but here we needed the tumor tissues to guide us, because peripheral blood data on its own, unfortunately, doesn't give us enough information. But with the guidance from the tumor tissues, we were able to now look back in the peripheral blood and see that if you look at pre-therapy, post-therapy, after first dose and after second dose of anti-CTLA-4, you clearly could see also this increase in the ICOS positive subsets, both CD4 and CD8 T cells. We then used ICOS as a pharmacodynamic biomarker of anti-CTLA-4, and at the time when we did this analysis, we actually only had 35 cancer patients, so the sensitivity was a little low. Specificity was very high. We now have over 1,000 cancer patients, and so the sensitivity is also greater than 95%. And so ICOS truly is the pharmacodynamic biomarker that we use for all anti-CTLA-4 therapy studies. We also wanted to understand whether or not ICOS could help predict for outcome, or at least be a prognostic indicator of outcome. And in the phase 3 clinical trial in patients with metastatic melanoma, Judd Wolchuk's group and our group had collected blood samples. And so in patients in this trial who we looked at retrospectively, we could see that those patients who had an increase in the ICOS positive T cell subset, those were the patients who had much better survival outcome compared to patients who did not. So that's the clinical data. And clearly the clinical data tells us that the ICOS ligand pathway is necessary for effective anti-tumor immune responses in the setting of anti-CTLA-4 as a hypothesis. And so to test that hypothesis, we actually went back to the mouse models where we could knock out ICOS or knock out the ICOS ligand. And so here you can see in the top line, these are wild-type mice. And if you treat wild-type mice that are bearing the B16 melanoma tumors with anti-CTLA-4, wild-type mice have an 80 to 90% tumor rejection and long-term survival. The same experiments done in the mice lacking ICOS or lacking its ligand, ICOS ligand, you can now see impaired tumor rejection and survival. And so this clearly indicated the importance of this pathway for anti-CTLA-4 therapy. So we asked the question, why is the ICOS ligand pathway necessary for optimal anti-tumor responses? And what we found was that ICOS is really, as I pointed out before, a family member of CTLA-4 and CD28. And all of these family members have the PI3 kinase binding domain within the cytoplasmic signaling to allow for PI3 kinase signaling and T-bet expression. So that was the hypothesis we generated. Most of you will think of CD28 as the important signaling for T-bet expression, for Th1 responses. What we were proposing here was that ICOS in long-term responses would actually be important in the setting of tumors because this is a long-term ongoing response. The CD28 may initiate, but ICOS may be very important for that later. And to test this again, we went back to the wild type on the ICOS knockout mice. And in untreated mice, we took T cells from the spleen. You can see the wild type and ICOS knockout mice do not have much in terms of expression of T-bet. However, after treatment with anti-CTLA-4, we took T cells from the spleen, tumor-draining lymph node, or the tumor-infiltrating lymphocytes. You can see the wild type mice clearly have an increase in T-bet expression, whereas the ICOS knockout mice do not. And here we're saying ICOS is important, but whether or not it's the signaling through the PI3 kinase domain is not clear because we're knocking out all of ICOS. So we went back to the ICOS-YF mice, and these mice have one amino acid change from Y to F. So they can express ICOS, but they cannot signal or bind through the PI3 kinase pathway. And here again, we can then sort from wild-type mice or the ICOS-YF mice, either the ICOS-positive T cells or ICOS-high T cells compared to the ICOS-low or ICOS-negative T cells. And if you look at this in wild-type mice, clearly T-bet expression after anti-CTLA-4 therapy is high in the ICOS-positive T cells. And in the ICOS-YF mice, you can see neither the ICOS-positive or negative T cells can upregulate T-bet. And I need to point out again, all of these T cells are CD28 positive. So it's not that they're lacking CD28. This is all ICOS driven. The third hypothesis that we had was that the ICOS ligand pathway can be targeted and developed as a combination therapy strategy with anti-CTLA-4 or other immunotherapies to improve anti-tumor responses. And I have to say that in full transparency, this is a company that we developed from this work to target ICOS. And to do that here, you can see, again, we went back to the wild-type mice. And in these mice, we could give a larger tumor volume and treat with anti-CTLA-4 alone, where you can see in the black boxes. And if I have a pointer, in the black boxes here, you have about a 20% response rate with anti-CTLA-4 monotherapy or with ICOS monotherapy alone. However, when you do the combination therapy targeting both ICOS and anti-CTLA-4, now you can have 90% tumor rejection and long-term survival. The same experiments done in wild-type mice were done in knockout mice, in the ICOS knockout mice, and you completely lose the ability to have this tumor rejection. And so I'm happy to actually say that ICOS antibody is actually in clinical trials, and we're waiting for data, and it's really fun to see your work go that far. So what about other tumor types? We just talked about melanoma and bladder cancer. And so people are thinking now that the reason that certain tumor types are responding to immune checkpoint therapy has to do with mutation load. Those tumors at the top of the graph here, like melanoma, bladder cancer, lung cancer, have high mutations. And because they have high numbers of mutations, they have high number of antigens that can be presented to T cells. And so probably the tumors that have lower numbers of mutations, such as prostate cancer, pancreatic cancer, or glioblastoma, those tumors may not respond to immune checkpoint therapy because they have fewer mutations. As an immunologist, it's hard for me to believe that because, T cells only need one antigen. They don't need 1,000. So we're struggling a little bit to understand this concept that it's only high mutation load that will respond. And so we're not quite sure that that's accurate. So we published a review article before that maybe high mutation load tumors have this scenario, where there are already lots of T cells infiltrating into these tumors because they have lots of mutations, lots of antigen, and so all you have to do is take off the inhibitory pathway from these T cells, and the T cells can do their job because they're already there. And then the other tumors look like this. They may not have the T cells in there, so if you try to block the inhibitory pathways, there are no T cells. So blocking the inhibitory pathway is not going to really get you much. And really what you need to do is convert the tumor from this cold environment to a hot tumor microenvironment where there are more immune cells, and then try the immune checkpoint therapy. And so we looked at different tumor types to understand if that's true. And you can see here in prostate cancer, it's actually one of the coldest in terms of immune infiltrates. Prostate cancer has very few immune infiltrating cells compared to other tumor types. And so we thought, can we increase immune infiltration into prostate tumors and then look at what's happening? And since we knew anti-CTLA-4 drove a lot of T cells into these bladder tumors that we saw before, we thought we'd try this too. So here again is another one of our clinical trials, which is in 20 patients, where we were just going to ask the question whether or not hormonal therapy, which is a standard of care therapy for prostate cancer, can be combined with anti-CTLA-4 to now increase the tumor immune infiltration into prostate tumors. And so we were able to now convert this cold prostate tumor microenvironment to a hot tumor microenvironment. Clearly, we went from very few T cells to many more T cells into the tumors in multiple patients. But what we weren't expecting was that there were other resistance mechanisms that would come into play. And this gets me back to the yin and yang of the immune system. You can't drive an immune response completely without expecting to see some sort of regulatory mechanism become upregulated. It's just how it all goes together. And so we hadn't thought that through completely yet. And so it was good to see that we're learning. And here you can see our gene expression data showed lots of regulatory pathways increasing, including PD-L1 and VISTA. And so when we looked at PD-L1 expression from our tumor tissues, this is a pre-treatment slide that shows very few T cells, and then post-treatment with a complete merge of all the different immune cells. We're looking at CD4 in green, CD8 in red, CD68 for macrophages in yellow, PD-L1 in white, and the nucleus DAPI in blue. And you can see in the merge now, you have all of the immune cells present after treatment. And the CD8 T cells are here, but now all of the CD8 T cells are expressing PD-L1. Macrophages are also present, but they're also expressing PD-L1, which is another inhibitory pathway. And so when we quantitated it, you can see it was significant in all of the patients between the pre-treatment and the post-treatment is in red, looking at PD-L1 expression. So the gene expression data correlated with the protein data. VISTA is a very novel and new inhibitory pathway. I think we're all familiar with CTLA-4, PD-1, PD-L1, but VISTA is relatively new, discovered by Randy Noel's group. And again, what we saw here was that the CD8 T cells express VISTA, but the monocytes that were infiltrating macrophages now were expressing a lot more VISTA. And again, when we looked at it statistically, we could see that VISTA was clearly upregulated in these tumor tissues. VISTA and PD-L1, though, represented two distinct inhibitory pathways in the tumor tissue. So we looked for whether or not you had mostly VISTA positive or PD-L1 positive or double positive cells. And what we could see was that there were very few double positive cells, and you had two distinct populations of the VISTA and the PD-L1. So again, the immune system is putting multiple different pathways in play. When you drive it to go on, you're going to see multiple different things come into play to control it. And just to show that VISTA is a potent inhibitory of human T cell responses, we took the T cells from these patients, and if you engage the T cells with plate-bound VISTA or PD-L1, you can see that, again, you lost interferon gamma and TNF-alpha as a result. This is with plate-bound PD-L1, this is plate-bound VISTA. And VISTA decreased the production of interferon gamma and TNF by these T cells. So what are some other resistance mechanisms? Because again, tumors are very smart, actually. They do come up with ways to evade the immune response. And that was what we found in some of our other patients. These were patients that we took from our melanoma cohorts who were either not responding to immune checkpoint therapy or responding. And this is just sequencing of the patient's tumors now. So what's different about the tumors that are responding versus those that are not responding? And what we actually found was that patients whose tumors were not responding or were resistant to therapy had copy number alterations in the interferon pathway genes. And this makes a lot of sense because T cells produce a lot of interferon gamma when they're activated. And if you now downregulate the interferon signaling pathway in the tumor cells, the interferon is no longer able to induce apoptosis of those cancer cells. And so we were able to clearly show that these patients who were the non-responders had a loss of these signaling pathway genes. And so we went back to the mouse models and we actually knocked out the interferon gamma receptor in a tumor cell line. And so in the top line, this is the tumor cells that are intact for all the pathways, and then in red, if you knock down just the interferon gamma receptor in the same tumor cell line, you now lost some of the responses that you saw before. And really what I'm trying to say is that the resistance mechanisms can be many different things that lead to resistance. It may be upregulation of these new checkpoints that we're not even aware of, or it may be that the tumor cell's adapting in different ways. And not only did we show the interferon loss, it was also shown by Tony Rebus's group, and they also showed loss of beta-2 microglobulin as another pathway, because then you can't stabilize the MHC for expression of antigens. So I'm just going to end because I want to point out how much work we have to do. So these are all the pathways that are in play, and we're still just focused on CTLA-4, PD-1, PD-L1. Today I talked a little bit about ICOS and VISTA, but there's still a lot more work to be done because we don't understand how all of these resistance pathways or inhibitory pathways are working. And I can tell you that there are hundreds of clinical trials ongoing, so we really need somebody also to help us make sure we understand how these pathways are working so we can correlate the clinical data and hopefully more translational research in this space. But more importantly, I think we're now in this realm where, as oncologists in my clinic, I'm no longer just talking about improving survival by a few weeks where everyone still dies. That's no longer what we hope for. We are here. This is clearly where we are with immune checkpoint therapy. We now have about 20% of patients who we would like to consider cured. They're coming back to our clinic for 10 years or more, so they're doing very well. And then the goal, of course, is to get it here. And this is where we are now in trying to understand these resistance pathways, trying to understand what these other inhibitory molecules are, and how to go about doing this with combination therapy. And I really think we can get there. It's going to take some work, but we're on the right path. So just to conclude, immune checkpoint therapy has certainly joined the ranks of surgery, radiation, and chemotherapy as a pillar of cancer treatment. The ICOS-ICOS ligand pathway may serve as another target, and that the clinical trials are ongoing. Multiple immune checkpoints exist and are dynamic in their expression, so they have to be evaluated in both pre- and on-treatment samples, because if you just look at pre-treatment samples, you think that there's nothing there, and then when you look after a particular therapy, you can see these changes, and that will help to guide therapeutic decisions that we make. Resistance mechanisms are going to involve expression of additional immune checkpoints or generation of tumor cells that lack certain pathways that prevent or evade the immune response. More importantly, I really just want to end with this point here. Pre-surgical and tissue-based clinical trials really do provide a feasible immune monitoring platform. They really allow us to study biologic effects within the tumor tissues that can lead to meaningful data that can be applied to the metastatic disease setting and provide insights into mechanisms. There are a very small number of patients involved in these trials, like I said, 10 to 20, but they give us a huge wealth of information that can then be applicable to many other settings. I think we really need to start considering how we do that more and more in different clinical trials instead of just being stuck in the standard paradigm, because I think all of us in this room need to understand more what's happening in human beings. We've cured mice for years. I really would like to know more and more what's happening in people. So with that, I'd like to thank a lot of people. This is always a team effort, collaboration, collaboration, collaboration. I'm grateful to all of our surgeons, our pathologists, our oncologists, our immunology team. Most importantly, I really do want to thank our patients. These are patients who can go directly to surgery. I mean, really, that's their standard of care, is to go to surgery. And yet they allow themselves to participate in these clinical trials where we learn something from them. And that I'm truly grateful, and I would like to acknowledge them. So thank you all, and I'm happy to take questions.
Thank you so much, Dr. Sharma. That was great, inspirational, I mean, really exciting and worthy of all the breakthrough awards of the year that this area was given. So your point about unleashing the immune system, I think, is really interesting. And presumably, if we knock out and stimulate and effectively, unleash, there's a downside. And so is there a way to turn it off at the right point, or how many can be combined when you take the ICOS agonist and the PD-1 antagonist or the other checkpoint inhibitors? How many is too many? Do you reach a point? And what would be that point?
So I didn't get to talk about the toxicities, but toxicities are real. They're really inflammatory toxicities, so dermatitis as a rash, or pancreatitis or hepatitis. And the good thing is when we've combined these agents, when we've combined anti-CTLA-4 and anti-PD-1, we see the same toxicities, maybe higher frequency, but the same toxicities. And the good thing is that when you learn to recognize what these toxicities are. If you see a rash or you have a patient with diarrhea, you automatically think colitis. When you recognize it and you treat it earlier, and the treatment is steroids for all of them, when you treat it earlier, we tend to see that those toxicities can go away and they can resolve, but the anti-tumor effects can continue. And this is and we're learning more about that too, because we think when we're giving the steroids, it's really preventing the expansion of new T cells that are towards whatever those antigens are, the self-antigens more, and the memory response may have already occurred towards the tumor antigens, because that may have been the first wave of response. But we're still trying to understand that. But I really think it's important that we are not afraid of toxicities, because again, these are patients who are dying within 6 months to a year. So as oncologists, we're not afraid of toxicities. I mean, come on, we came up with stem cell transplant. Are you kidding me? This is nothing. These are outpatient therapies. We give the patient therapy for 90 minutes in an outpatient setting and send them home. So yes, there are toxicities, and I don't want to minimize them, but they need to be recognized and they can be treated. I still remember the days when we treated with cisplatin chemotherapy. I don't know how many of you may remember, but cisplatin was said to be the worst thing. It caused kidney failure and neurotoxicity. And now we give it like water because we know how to give it and we know how to recognize the toxicity. So I think we will get there, but it will require some education. But I do think the good thing is seeing that these toxicities are now easier to recognize, easier to treat.
Great, thank you. Adriana.
Thank you for an excellent state-of-the-art presentation. I'm an endocrinologist, so my question is related to Monica's. Actually, what we see is our cases of hypophysitis and primary adrenal insufficiency, autoimmune diabetes even, and they don't go away. I mean, of course, we can replace, we can treat. We don't even use high-dose glucocorticosteroids in these situations. So the question is whether through your mechanistic studies you have come up with a way of associating the toxicities with the response to medicine. Is there a correlation between the degree of autoimmune toxicity and the anti-cancer response from this immune checkpoint inhibitor?
So earlier, when we had a few hundred patients, there were some publications that stated that. In an analysis of over 7,500 patients, we don't see any correlation between toxicities and response. We have patients who respond without any toxicities, and we have patients who only develop toxicities without any response. So that I wouldn't say that's a correlation at all. I think there are definitely antigens that are shared, that you may see some type of responses between the tumor and, for example, brain or colon and these kinds of things. They're also in the colitis setting, depletion of regulatory T cells in the colon that people think are also related to these toxicities. But there are multiple mechanisms that could be leading to them, and unfortunately, they're not linked to the tumor rejection.
Thank you.
Gary?
So you can't study everything, but you didn't mention Th17 cells or a role of those and with the ICOS and all that. So you were looking at the Th1s. Do you think there's roles for these other T cell subsets?
So we did look at Th17 because the mouse models clearly show Th17 cells are important in the anti-CTLA-4 treatment. In humans, we never saw that. It was always Th1, predominantly Th1, very few Th17, did not ever reach a statistical significance in differences across hundreds of thousands of patients now, but in the mouse models, I will tell you over and over, Th17 comes out. We don't see the same thing in humans, so that's why I didn't present it.
Hi, that was really fantastic. You mentioned between different tumor types, such as prostate cancer and melanoma, the differences in mutation rate and immune cell infiltration. I'm wondering about within a given tumor type, like melanoma, the responders versus non-responders, what the predictors of mechanistically of response are.
And that was in the last slide. That's what I was showing in the interferon pathway. So that was melanoma, responders, non-responders.
So, but do they have the immune infiltration within? Does that make a difference?
No,
or
they
just
tend to
interference
have the
support?
same immune infiltration. So in melanoma, they tend to have the same immune infiltration, whether they're responders or non-responders. I will tell you that if you take out the worst non-responders, meaning they progress right through therapy instead of just being stable disease, and then progress, and take out the best responders, meaning that all the tumors go away, then you will see big drastic differences between the immune infiltration. But, it's a conglomeration of patients, and so when you start to put them all together, some people consider if you've had stable disease for a year, that's a response, okay? It didn't all go away, but so then it gets a little bit trickier. But if you do take the two extremes, yes, the immune infiltration can predict.
So will immune genomics of the cancer, is that going to go away? Are we not going to need to look at the mutations and such things anymore?
No, I think you're going to have to look at combination therapy, so you do need the genomics. So, for example, do you need an EGFR2 targeted, agent combined with an immune checkpoint agent? And so I think those are things, the combination between genomic medicine and immunotherapy, I think, will be very important.
Any other questions? If not, thank you very much for a wonderful presentation. The next thing I wanted to do, I failed earlier to acknowledge, as we were talking about the transition in the journal, we honored Dr. Ploth, but I want to also mention Mike Yulian, who has been a long-standing supporter and part of the editorial team for the American Journal of Medical Sciences, and I want to just applaud his years of service to the journal as well. So thank you very much to Mike Yulian. I'd like to quickly call up Joan Kemp and Pamela Schmidt, who are part of our administrative team, who have made my year wonderful and smoothed every pathway. Every crisis was dealt with speed and efficiency, and they've kept me going. And I just want to give them each a small token of appreciation, some nice meals at some nice restaurants. So thank you so much for everything you do. And I'll mention that Pamela represents our new alliance with Tulane CME office. She is actually part of our administrative team. The CME office has been long involved in our meeting, but now she's actually serving with Joan in an administration role with SSCI, and it's been a great year. Okay, it's my pleasure again to proceed with a few more award presentations for young investigators, and then we will move on to the Founders Medal. So the next announced award recipients are the SSCI Young Investigator Awards. These are for the best of the best. We have great presentations here at this meeting, but these people have really risen to the top in terms of the review process. So they've been selected based on the merit of their abstract submissions to the meeting. And our first place recipient is Mohammed Koss from the University of Alabama at Birmingham. And if you're here, please come up and accept your award. Congratulations. Big honor. And our second place recipient, I had nothing to do with this, but she's from Emory University in my division of infectious diseases, Valeria Cantos, her abstract was on HIV risk behaviors among immigrant Latino youth, a public health priority. So congratulations, and come up and receive your award, Valeria. Congratulations. All right, and our final third-place recipient is Dr. Nindi Jain from the University of Tennessee Health Science Center, and this is abstract temporal trends and clinical outcomes in patients with uncontrolled diabetes and related complications. So congratulations, Dr. Jain. Very good. Thank you. We, in another venue later this morning, are going to award an award to the best abstract in the Society for General Internal Medicine. This is an SSCI-sponsored Young Investigator Award, and I'll be presenting it in their session later this morning. So congratulations to Alex Galloway, who was the recipient of that award. And now we are going to present our final awards before moving on to the Founders Medal. We have a competition. It's a relatively new competition among the poster presenters. Last night, a group of judges went around and visited the top 3 posters and had each one of the individuals present their poster. It was my pleasure to join the judges, but I was just there to watch and to hear, and it was very, very nice presentations. We had a pulmonary critical care fellow, and we had 2 medical students who had done summer projects. So it's my pleasure to announce the number 1 first place award recipient for the poster awards, and that is Lucian Martz from Emory University. Make sure it's you. Yes, it's you. Congratulations. Very good. He did a great presentation. I should also say that each one of them had wonderful mentors, and I think that's what this meeting is about. Our second place recipient is from the Medical University of South Carolina, Christina Kurse. Please come up and receive your award. Good job. Congratulations. Thank you. Thank you. And our third place recipient is Austin Castillo from the Texas Tech University Health Science Center. So congratulations, Austin. Come on up. Great job.
Thank you.
All right. So congratulations to everybody, all our award recipients. It was a great job. Okay, so now it is time for our SSCI 2017 Founders Medal presentation. And to kick off that presentation, I'm going to invite Jesse Roman and Sushma Cribbs up to the podium to introduce our 2017 award recipient.